Yes. Good evening. I hope you have downloaded the files. In that mail, there are two mails, one with QTL outputs and one Word file with name methodology. And in other mail, some several scripts are there. Yeah. So that QTL interpretation we will cover tomorrow. So today we will start the session with alpha lattice. So just open the R code of alpha lattice and simultaneously open the data. Yeah. So here you can see, am I audible?
Yes, sir.
Yeah. So in alpha lattice data, here you can see their first column is replication, second block, third is treatment, and from fourth column, data is there. So here you can see first is replication and treatment. Here in between one extra column is there, that is block. So can anyone tell me what is actually this alpha lattice is? How to analyze that is different thing, but what is actually alpha lattice is? Like in RBD, we will take some number of n number of treatments, we will replicate it 2 times, 3 times, and we will do analysis. So likewise, what is actually this alpha lattice is?
Hello, sir.
Yes.
So alpha lattice is an incomplete block design, and
Okay.
this is used when you have large number of treatments that cannot be accommodated in a single block. So here the sources of variation here you have the rep and you have the block that are in each of the replications. So you use it
Okay.
when you have large number of treatments.
Yeah. So likewise, here you can see there are two replications, right? But within each replication, you have two blocks. So likewise, see, there are 6 treatments. So 6 treatments divided into 2 blocks. So likewise, you need to arrange the data, number of replications, blocks, and treatments. So actually here, while analyzing the data, each replication is divided into some number of blocks. And by using that, we can do the analysis with large number of treatments. So this large number of treatment means it is more than 200, 300, even 500 to 700, more number of treatments. If you will see the script, so this is the code. So just first I will set the directory. Then Just run this library commands. If any package is not there, just install it. You can import your data set by this command line. If anyone is having their original alpha lattice data, they can analyze. Just they need to change these names. So then after factors, you need to define the traits. Then analysis with LSD. That is a complete one up to this. So then at last, your all plots and files, it will be there in your Excel. So see. So like this plots, this is with LSD. Then this distribution of threads, thread distribution by treatment, box plots will come for each and individual thread. Then again, this residual versus fitted, just like we have seen during FRBD and split plot. So like this images will come. And as far as the data are concerned, so this is mean. Then these are the LSD values. You'll get Excel files with the residuals. So this is alpha lattice with bar plots, box plots. These are the bar plots. So this is, so that same earlier I have shared one script with you. I think it is through mail only. So that script you can use for analysis of variance, because I think here ANOVA is not there. So analysis of variance you will get from that old script with same data format. So this script mainly it will focus on this graphs. So the same data format with that script, you will get the outputs. So this many things you got, plots. Or if any error is there, just tell me. Or if completed also, tell me. So that I can proceed. Okay. Second thing is this is full diallele. So the second thing is full diallele analysis. So in this case, we are using this data. Yes. So now tell me what is meant by diallele? What is diallele analysis? Yes. What is diallele? What is line into tester? Yeah. What is meant by diallele?
Hello.
Yes.
So it is a kind of a breeding strategy or approach in which the parents are crossed in all possible combinations. This is to estimate the general combining ability of the parents and also the specific combining ability of the progenies.
Okay. What is meant by direct and reciprocal cross?
Yeah, reciprocal cross is that is just a concurrent. You cross, you make selection, and you put it back to the crossing block.
So just see this data set and tell me whether direct and reciprocal both type of crosses are there or not.
Yeah,
Yes,
reciprocal, at least one could be
yes,
male.
sir.
You can
There
use
are
one as a
reciprocal
male, then
classes.
use the other as a female. Then you can also use the other
So
as a female
see,
and use,
Yes, both
yes,
this
are here.
yes.
this third line, one cross with two. So what will be the reciprocal of this?
2
2
and
is
1.
2.
So here it is 2, 1. So it is 1, 4, 4, 1, 2, 3, 3, 2. So that means here we have arranged the data with that data is containing direct as well as reciprocal crosses. And then total number of applications are 3. So if reciprocal crosses are there, so that means which type of diallele is this?
Full.
Full
Full
dialysis.
diallele or half diallele?
Sir, full
Full di-
dialysis.
And
full
what about half diallele?
Sir, in half dialyl, reciprocal crosses are not included.
So like this, you need to arrange the file. And if you will see, you need this all these packages. So just instead of library for each and individual, you can run this command. So it will come as a library. After that, we need to use DDATA file name. This file name DDATA. Then we will define the trait. This time here we will create the output folder for output file, and then here we will run for each and individual character. Something is wrong. So here this folder will come. So here you will get the ANOVA for each and individual character. Then secondly, you will get GCA. See, representation of GCA for that particular trait. So it is for each and individual trait you will get. Then here you will get heterosis on mid parent and on better parent. as we have not given the values for standard heterosis, so we will not get the standard heterosis. What is meant by standard heterosis?
Compared to a standard check, sir, commercial variety.
What?
Standard.
Okay. Yeah, and what is meant by this mid-parent heterosis means what?
Average heterosis.
Average basis.
How we will calculate?
Mid parental value by calculating mid parent.
How will calculate mid-parental value?
Average of both the parent P1 and P2.
So here that also you can see this is parent 1, parent 2, and then average will come. So this is better parent heterosis, and as these values are not there, so this will not come. So likewise this heterosis, and for that, for heterosis also you will get this type of heat map for each and every trait. So here you can see either the heterosis is positive or negative.
Sir, in case of like flowering,
Yeah, my
leafage,
question is that only. Wait. My question is that only.
yes.
So see, here you can see negative values and positive values both. So generally in any crop, just tell me which type of which type, either negative or positive, which type of heterosis is desirable.
So generally positive.
Any characters are there where negative heterosis is desirable?
Yes, sir.
Plant height,
Early
Plant height, days to flowering,
early
plant height
moisture.
moisture,
is based on maturity.
Okay.
earliness,
That you can say, like in other words, days to first picking or days to maturity. If days to flowering is will come negative, then that maturity and for first picking, that's it, for that traits also it will come negative value. So this here you will get the ANOVAs. This is your mean sum of squares. For dialyl analysis, and these are the heat maps. Yeah, and you will get one text file also. This is ANOVA, GCA, SCA.
Where is the code for that heterosis, sir?
Same code. What we are using. Same code yielded this. How many files? 60 files.
Diallele only, sir. From diallele that.
Ah. So here you will get heterosis. So better to use Excels, and then these are the SES. These are the SCAs. Can anyone tell me what is the difference between additive and non-additive gene action?
In additive
Hopefully,
gene action,
uh,
the cumulative effect of each individual gene. There is continuous variation.
okay. And what about non-additive?
Dominance effect, sir.
The
Okay. So
additive is
then,
fixable.
uh,
Fixable. Additive is fixable.
okay. And what about non-additive?
Dominance and it is
In other
process, sir.
words, I will ask that question.
Uh,
If your specific combining ability variance is more than general combining ability variance, then which type of interaction will be there?
So non-additive.
Non-additive gene interaction.
So that means, what is the meaning of that?
When
Hmm?
GCA is more, sir,
Hmm.
it shows the additive gene action, and we
Okay.
can go for selection for the improvement
Okay.
of that. And for non-additive gene action, when SCA is more, it will, the non-additive gene action will be there. At that time,
Uh-huh.
selection will not work, and we have to go for heterosis or other breeding methods.
Yeah. So the conclusion for non-additive is, as just now Madam told, like we need to go for the breeding methods related to development of hybrids, because there will be no scope for selection. Then better to go with heterosis breeding. So that is the use.
So, so how will you, sir, identify this non-additive gene action where it is happening?
So see, actually, whenever your SCA will be more than GCA, so the interaction will be non-additive. So this additive, non-additive will be there on the basis of your GCA and SCA. When you will arrange all these tables into that particular format,
Sir, any crop, sir? For any crop?
concept is same.
Okay.
Concept is same. As we are well aware, from GCA, we can select parental lines. And we will test this specific combining ability. If that ability will come, means that interaction will come as a non-additive, then you need to follow the breeding methods like heterosis breeding, because the scope of for selection will end there. As far as when your GCA is more, then there will be the scope for selection. Like means it is not like if you are testing hybrids and you will get additives in action. So this combination is wrong. If you are saying whatever you are testing for specific those are hybrids, then that interaction will be compulsorily non-additive. If you have made the crosses, actual crossing you did, then that interaction should be non-additive.
Excuse me, sir.
Hmm.
Sir, if we go and see the result, what we analyze, like the
Hmm.
result of TSW, like it's a seed weight, something, test seed weight. So in this analysis, there is GCA and SCA, both are here for different crosses. So if we, there is two things like GCA 1, GCA 2, cross for cross 4
Yeah,
into
for
2.
each line it is like 1, 2, 3, 4.
Oh, okay. So GCA also like GCA 1, GCA 2. Was the why it's saying GCA 1 effect and GCA 2 effect? Is there some difference in GCA 1 and 2?
So here
So
you can see.
I'm checking the text file what we got after the diallele data analysis, and in this is saying like GCA 1 and GCA 2 for
So
the cross.
it is based on this cross 1 and cross 2.
Okay. So it was not like the cross 1:1, 1:2. Okay. So GCA 1 is for cross 1 and GCA 2 is for cross 2.
Yeah,
Am I
it is
correct?
already mentioned. It is like, say, here it is mentioned 1,
Okay,
and
sir.
then here it is mentioned as 2.
Okay, sir. And in this data, if we can see like there is just one positive value in cross GCA 2,
Now,
we
if
just
you
get
will
one
if you will apply the significance and non-significance, so
yes.
after that you need to interpret it. Though it is either negative or positive, but you need to interpret whether it is significantly positive or significantly negative.
Okay, sir. So how can we
So,
know like it's a GCA is more
as
and
this
SCA is more by
as
looking the data?
as this trait is field grains per plant, so here in this case, field grains means here only first line will get significant.
Yes, because it's the we have positive heterosis. We need here positive heterosis. Okay, sir.
And for that, what we have told now, I will go back once. So for that variance, you need to go for that line into tester. Where we did that. That day we did na line into tester analysis.
Yeah, we perform, I think, one
There,
day. We should have.
there you will understand easily. There we will get the genetic parameters. So from that, there you will get wherever you will get the SPA variance.
Okay, so we have to analyze
CCA.
the line into tester variance, and then we got the
Yeah,
different
so
genetic
from because
component.
interpretation, because see, there are different methods like diallele. In diallele, you will get full diallele, half diallele. In full diallele, we will generally include directs and reciprocals, and from all those, we will interpret it. We will just select which is significant for particular character, and then we will proceed. But in case of line into tester, what will happen now? Each and every line, yeah, that lines will get a chance with each and every tester. So from that, you will get GCA variance and SCA variance. So if your SCA variance is more than GCA in line into tester, then you can interpret it as a non-additive gene action.
Okay, sir. So first we have to we have to run this command as well. That this is and the line.
Because generally now in this research and everything, we'll generally use lining to tester. Full diallele will not use.
Okay, so we have to run that script, the line into tester script in this data
That
only.
that data format is different.
Okay, yeah, line into tester is different.
I'm not getting where I kept the output file where I kept. I don't know. Wherever I will get that, I will show to you. No problem. So this is full diallele. Then actually one script is there. I don't know whether this script will work on large data set for generation mean, because this generation mean, as yesterday I told you, it is recently added to R, but still we will try. So generally, how many generations will be there in GMA? Yeah. So here you can see in generation mean analysis, we will handle 6 generations. So this side took only one thread. So this is all 6 generation and this is the mean value. Likewise, you can modify that according to your data. So then we will run this library. And then I will annotate this file. If you don't want to annotate, then change this extraction commands. Okay. Then here we'll calculate all the effects. Yeah, I think it is working. No problem. So here we'll get the effect values, gene effects, and this is generation mean for that particular trait. This is generation mean for that particular trait with 6 generations, P1, P2, F1, F2, BC1, BC2. And then these are the gene effects. So likewise, first is additive, then A into A into D, and D into D. So here you can find that is dominance into dominance effect is more.
What does it mean, sir?
One minute. I'm just one second. Yeah, so just check your mails. Each and every file is there only in that zip folder. And while copying the code, just see where the bracket is started, where it is ending. So if we will copy paste in between, so that will not run. Yeah. So as far as this diagram is concerned, here you can see on x-axis, you will get the different type name of different types of genetic effect, like A into A into D, D and D into D. And on y-axis, effects value. That is magnitude and direction of this genetic effect, either positive or negative. Okay, so here you can see this effect value for dominant and dominant, as I told, it is 44 in positive direction. So that means the interaction between the dominant alleles for that particular trait, it is positively influencing for the improvement or development of that particular trait. So, if suppose this trait is plant height and dominance effect is value is 44. So that means whatever the dominant alleles are there, or whatever the dominance concept in between the alleles. So whatever the interaction will be there, so that interaction is positively influencing the improvement of plant height in all the 6 generations. Whatever the research we are doing, so this interaction between dominance alleles that are influencing positively. So vice versa, additive effect value is minus 50. So means this additive interaction significantly reduces the expression of that particular trait. So means out of all these 5 interactions, you can conclude like additive into additive and dominance into dominance, they have strong influence on that particular trait, respectively on negatively and positively. So this A into A, it will reduce the trait expression, and this D into D, it will positively influence the trait improvement. So likewise, this interpretation, and then graph. Yeah, so this graph you will get here only. Already it is here in your working directory, both the graphs. So whatever the multiple traits will be there, so this traits, so for each and individual trait, this type of graph is there. And optional to this, yesterday I told that is GMA package, GMA R package. So that package also you can try. So here last, if you can see the command line, here you can give this command, as many publications are asking the DPI level of image is 300. So this command you can give so that you will get the image in 300 DPI quality. Like this. And this is the estimated genetic effect. Yeah, madam.
Excuse me, sir. Sir, I have one question. Like when there is a fixed population and germplasm, we go for QTL mapping or reverse. But if
Mm.
this we have this kind of population where heterosis, we are doing heterosis breeding and something, then what should be we have to do for molecular breeding? I think. How can we do analysis for molecular breeding if we
I mean,
find try
which
to
type of population you can you are asking?
No, sir. I'm asking like we study like the population that have the fix. We can fix the trait that is due to additive gene effect. So we go for QTL mapping like rays, and in the germplasm we go with jivas. But if we have this kind of population and we have to go with the molecular breeding, then what should
So
we have to
simply
do?
say, if you are developing particular hybrids from parental lines, okay, so just uh, after development of hybrids, we will plant those hybrids, and after one season from the data, we will record the data and then we will analyze. So instead of that, by using some markers, you can test the hybridity of those hybrids, whether it is really hybrid or not. You can compare by using some sets of markers. You can screen the parents as well as the hybrid.
Okay, sir.
So it will save your time. So
Okay, sir.
if you have developed some 500, 700 hybrids, and after screening, you came to know that only 200 hybrids are the actual hybrids. So ultimately, it will save your time and space and money.
And also the hybrid will be breakdown after different after passing through different generation. It will be so we cannot fix that hybrid with any molecular breeding aspect and anything by applying. Is this possible?
Yeah. Look, what you are means breakdown of hybrids you are asking,
Yeah.
or
So
what?
after some generation, it will break down, but like it will be not. We cannot fix it.
So means you are asking from F1 to F6.
Yeah. Is this possible? Like, yeah. Yes.
So actually, means see, actually what the industrial, especially industrial people I am asking. So doctors have correct me if I am wrong. So generally what the people are breaking down the hybrids. Means uh, mainly their hybrids also they are breaking down for development of lines. But just they are not using any molecular tool. Totally, they are following the field trials only. On field level only, they are doing these things like F1, F2. In F2, they are selecting the maximum population, and from F3 onwards, instead of selection, they are going for rejection.
Okay. And then they
But as
select
far,
the it's
yeah,
like
and
talent.
whatever the best, it will remain at last.
Okay, sir.
Yeah,
But sir, if
because
we go
uh,
work with the tree crops, like horticultural crops, where the life cycle is longer, in that part, it's very hard to take one generation and do because
yeah,
sometimes.
failed. So in that case, you can screen that with some sets of markers initially.
And then go with the somatic method to
Yeah,
tissue culture method to propagate it.
yeah. So generally, that tree crops having long 2 years, 5 years generation time. So generally, that somatic method is there. Means at research stations also they are following wherever the facilities are available.
Okay, sir.
Hello, sir.
Ah, madam.
Uh, I want to put some points for fixation of heterosis. We can go for in vegetatively propagated crop. Actually, we F1 is propagated. F1 when we will develop, it will be propagated through vegetative parts. So it will be
Yeah,
naturally. And in sexually propagated crops, apomixis is one of the option, but it is not found in all the crops. It is difficult.
yeah. In majority of crop, that is not found beneficial yet. They are still working.
Ah, yes, somatic hybridization, as ma'am said, that is also one of the option.
Actually, in major crops like chili, that is still it is under process.
Yes, yes.
For chili
Okay.
also, almost all researchers they are working on field level only.
Ah, yes.
Chili and even okra, where the genome size is very long, very large, very large. So in that case, this kind of methods it will not success easily.
Okay.
So you have to come to our basic level, that is field. So still in this lot of technologies are there, but still at some or other situation you can't ignore the field. Field, you have to go to field for certain aspects. Because as many people are working on breeding plus biotechnology, so I will tell you one thing that I was worked on marker assisted backtracing in Chile. One project was there. So as still as I don't know whether everyone is aware or not, anthracnose is most severe disease in capsicum. Anthracnose, powdery mildew, but still not a single marker is there. Validated marker. Identified markers you will find many, but not a single validated marker is there. No one is claiming that, see, this marker is there, you can use for anthracnose resistance. Even for restoration, like ABR lines are there, as in capsicum, you will find both lines, that is genetic male sterility as well as cytoplasmic genetic male sterility. The markers are there, but those markers are still not validated because of large genome size of chili. And we have lot of variation in species. Like capsicum annum is there, capsicum baccatum is there. So likewise, variety of species are there. So those chili is topmost, one of the topmost vegetable crop after okra. Okra is also having same situation. So that's why industrial people now started that GMS based okra hybrids. That GMS line was developed in IIHR. So that line, so for now, industrial people are concentrating more on GMS based okra hybrids. So that some or other situation is there where still molecular breeding is not successful. It is still under trialing stage.
Okay, sir.
Yeah. So just it, we'll come back on track. So now our next is the clustering methods. As one clustering method we have seen, that is our Mahalanobis. Now there are three methods are there. That is, one is k-mean clustering. Second is hierarchical clustering. And third one is fuzzy method. Actually, as far as in our crop improvements experiments are concerned, we will go with Mahalanobis. This fuzzy method, generally in soil science it is useful. They will follow this fuzzy method. And then this K-means clustering you will find in some crop improvement papers also, but not that frequently. Because frequently we will go with that Mahalanobis distance. And for all these 3-4 codes, we will use the data, our old data set, that is D1. First, we will go with this K-mean clustering. I will clear this everything. Okay. So here you can import the data through command line. Then check the structure. STR means structure. Then as we have included the replicated data, so this command will calculate the mean. So here you will get the mean. So then here we will remove the genotype column. So here our method is elbow method. So there are 2-3 different methods. So it is so this is K-mean clustering. Actually, this is the it is also called as unsupervised machine learning algorithm that is used to group the data into cluster based on similarity. Here, if you will see the command line, here we have given the number of clusters. So, in other way, we have estimated the number of clusters in our Mahalanobis distance. But here we have given the actual how many clusters we want. So that is one difference. After that, here is, so here you will get the cluster. Which genode, your which genotype is there in which cluster? Just you can sort this. Yeah. So this is completely your cluster 1, then 2, and then 3. Then like this, it is your cluster diagram. This is k-mean clustering.
Where is this k-means cluster data file? It is not there in that clustering zip file.
D1, 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 D1. It is there. This is the clustering folder, and here it is D1.
No, sir.
Yes.
Yeah, sorry. For the k-means clustering, so the diagram, yes, yeah. So is there a way to identify from the table we have seen the individual sessions that are in each of the cluster? Here, is there a way to identify those genotypes?
So you are asking here, you need numbers.
Yeah, yes, yes.
Okay, I will update that. No problem. I will see this. I will edit it. I will send. Because in tables you can see easily. Clusters are there. So that thing, when we'll go to hierarchical clustering, I think here we will get. Yeah. So here you will get the numbers. Lock folder. So again, you need to go to that same location. What you did last time. So same thing you need to repeat.
So why is it coming again and again?
That is the drawback of some packages. I think you run PCA that time again. That time these things came, na?
Yes, sir.
So same package here. Why you are installing this Factor Extra? It is already there, na?
Yes, sir.
So then directly run library. Don't install this again. Run library.
Okay, sir.
Thus, sir, this graph is similar to like PCA, no?
No, no, no.
Like dimension 1, dimension 2, and
Yeah, that actually see this clustering, PCA, diversity, it is everything all about the diversity. That ultimate goal is diversity only. Though these types of representations are different, as from D square we can group the genotypes, but ultimate aim is diversity. So you will get this diversity. That is here instead of PC1, PC2, here you can consider as a factors. So here your factors are genotypes and replications. So it is actually two components only, but here as we are clustering of the genotypes, that's why instead of PC, here you can consider like factor 1 is giving 23 and factor 2 is giving 21. So as this is clustering, so we will not interpret like about the diversity by factor 1 and factor 2. Our main aim is clustering of the genotypes. So here also if you will see, here also it is there.
Excuse me, sir.
Yeah.
Sir, in this silhouette method, we get the highest peak on k equal to 6. So why are we getting only three these clusters?
Where?
Sir, the previous one, the graph silhouette method. Yes, yes, sir, silhouette method. Sir, here k is equal to 6. So doesn't it gives the optimal number of clusters? But here
That's
only we
why
are
I
getting.
told. That's why I told. Here in K-mean cluster, we need to write the number of clusters. Here I wrote 3, so that's
Yeah.
why 3 is there.
Okay, so
So as per
according
the
to
graph,
this
you can
graph
write
only.
6. Yeah.
Okay, okay.
Here in K-mean clustering, you can change the number of clusters. So actually, optimal number is 6. So up to 6, you can write. I don't know this fuzzy method will work on this data. Just once try whether it will work on this data or not, because I think it needs more factors. This will be useful in more in soil science.
Sir, can you please check query with number 905?
Again error. This one, no? 1 minute. I
Group
will check
5.
where it is. Library structure. In K-mean clustering, na?
Yes,
Okay,
sir.
structure
Step
DTF
third, sir.
mean. What is the problem? Data mean. So how you imported the input file?
Sir, from that side tab, not through the code, sir.
Okay. So then first you annotate your file name with the data and then run.
Okay, sir.
Ah. Or else change the instead of data, write your file name here once and then try.
Okay, sir.
Either way you can do. Yeah, same thing. What I told for you also, Anjali.
Sir, can you please revise annotation method?
Okay, sir.
Simply see. So now as your, what is your file name?
Sir, D1.
Okay, so it is data, simply like this and write D1 and press enter.
Okay, sir.
If you will press enter as D1 is not there, in your case it is there, so it will work. So just here you can try this last method, fuzzy.
Sir, in the code for this k-mean clustering, in the step 4, you have given that remove gen column, मतलब genotype column. So, what does it
Yeah,
mean?
so that means this is not numerical. Just we want the data. We
Yes,
want the
sir.
quantitative data. So for that, we have removed this column, genotype column.
So, sir, how, like, in the representation, how we are understanding that it is removed?
So after removing, ah, after removing, you will get this file. And your next command is on data underscore numeric. If you will check, so this is data underscore num. So the same file, it will be there every time. So from here, data scaled is there. So we are taking the same, the file name generated from step 4, we are imported into step 5. Separate file will come, data underscore num.
Okay, sir.
Right into tester. We'll run it again. No problem. Yeah. So for that, you need this file LT data with first column replication, second column line, third column tester, and fourth column is the character. And this two-line script. First, I will upload the data. So here in case we are using the package Agricoli. And here output with our file name is not L into T, it is LT data. Then line into tester model, replication line tester yield. If you have more number of characters, you need to edit. Yeah. So likewise, here you can see it will give you the analysis of variance. After that, it will give you the GCA effect, line and tester. After that, it will give you the SCA effect, 1 into 678. These are in horizontally. So then it will give you the standard errors. So this I will come last. So then these are the proportional contribution to the total variance. What is the contribution of line? What is the contribution of tester? And what is the contribution of interaction? And the thing what I told you earlier, these are the genetic components. So here you can see
Excuse me, sir.
yeah.
So this LT script is available in that zip folder because in my folder it is
No,
not
no, no.
there.
Earlier I have shared. I don't know from where. I don't know when I shared, but it is already shared. Here just now I copy pasted.
Excuse me, sir. If it's possible, could you please share the code in WhatsApp group for line to tester?
Yeah. 1 minute, 1 minute. I will share.
So for K and hierarchical clustering, the input file is same. Hello, sir.
Yeah, for hierarchical, if you have any type of mean data,
Mean data.
or you can, if it is there, that will be easy. Any mean data. Yeah. So here you can see the genetic components. So what I was telling you earlier, the SCA variance and GCA variance. So here you can see the average GCA variance is 2.68 and average SCA variance is 411. So what is this HS and FS?
Half SIB and full SIB.
So which one will be useful for GCA and which one will be for SCA? What is half SIB? What is half SIB and what is full SIB?
Half SIB is when one of the parent is unknown. Maybe you have the female, the male is unknown. Then full SIB when both
So
male.
that means whichever individual is there, that is sharing only one parent.
Yes.
Yeah. And what is full SIB?
Both parents.
Yeah. So full SIB both parent means in our case, line and tester both, line into tester cross. That hybrid is sharing both the parents. And in case of half SIB means it is sharing only one parent. So that half SIB is used to estimate the additive genetic variance. So additive genetic variance means SCA. And this full SIB used to calculate the total genetic variance. So that total genetic variance will include
Additive plus dominance.
yeah. So total variance it will include additive with dominance. So from this data, you can conclude about that non-additive gene action? So likewise here, this is your additive and this is your total, that is additive plus dominance. But if here you can see majority of variance is contributed by dominance, so that's why this value is on the higher side. So this GCA is 2.68 and SCA is 411. So that means though the total variance is contributed by additive and dominance, as dominance is one of the non-additive type, so that's why from these two numbers we can conclude that the gene action is of non-additive type. So likewise, if you have multiple traits, so you can calculate for those multiple traits. You will get the same values right from ANOVA to contribution of line test. So this is line into tester. So everyone got these values?
Yes, sir.
So to understand this in more practical way, if you want to calculate it manually also, one biometrical book is there. I think author. I think Nadrajan is there, if I am not wrong.
Yes, sir. Nadarajan and Gunashekaran.
Yeah, Nadrajan book is there. That book is completely consisting of calculations only. You can easily use that book and you can do manually. Think like we are in 15th century and do it manually. If you will do it manually, you will never forget. That guarantee I can give to you. If you will do even if you will do the manual calculation of RBD, so anyone is there who did the manual calculation of RBD during masters or PhD? Manual, literally manual.
Yes, sir. We have done that.
Yes, sir.
Yeah. So whenever you will do, you will never ever you will not forget. Right from correction factor to analysis of variance table. You will never forget anything. So from that RBD, you can calculate this variability parameters, GCV, PCV, heritability, everything. Because whenever you will do manually, that everything that full concentration we need. And once it will fit in your mind, that will be there forever. This heterosis also you can calculate it manually, but now this is our, we are running from morning to night, we are running here and there, supervisor, boss, 9 to 5. So our lifestyle is becoming like that, so we can't do anything. Okay, so now half dialyl is pending. So half dialyl we will see tomorrow. And one more thing I will show you quickly. Yeah, that one more point is that QTL analysis, it will take a lot of time. So for quick analysis, if you want to say, just use 10 odd lines, 10, 20 odd lines and 30 odd markers. You can delete that file, other data, and you can run it from ICIM. Here I kept that output. Yeah. So here you can see, this is the methodology I have given. So here for filtering also I have given. Then if you want to construct only linkage map, so this procedure is there, and QTL mapping also. This data already you have. It's, I think, wrong file I have shared. 1 minute.
Sir, I'm getting error in that alpha lattice data script. Can you repeat that?
What is your number?
Last 7837.
7,
Sir, I have
7.
not sent you the screenshot.
Yeah, just share it. Let me see.
Okay, sir.
Yeah, so like this, see, like this output files will be there. Like this is static.
So your R studio is visible.
Ah, 1 minute. Yeah. So like this, output files are there. So this one by one interpretation we will see tomorrow. These are the chromosome positions. These are the LODs and these are the Ps. This linkage map is there, but it is not with LOD score. So I will try. I will search in my system. Somewhere we will get. Like this, plots will come. So this will complete tomorrow along with half dial. Okay. So meantime, I will prepare one data with 4 or 5 characters. So just try with this dial and align into tester. Try this analysis. Yes. Yes.
Excuse me, sir. Sir, I'm not getting how to annotate for this generation mean data. Can you please check? I have
Generation
sent you the
mean is
screenshot. My number ends with
okay. Error you got,
yes, sir. Sir, I'm not getting how to annotate the data in the first step of create data set.
So annotation is simple. What is the your name of your input file?
So generation mean data only.
Okay. So simple. Same copy paste. Whatever.
Sir, it is showing error.
No. How? Where is the error? Show me. Wait. You have already shared, no? So you need to set your this generation mean data is there in your clustering. Have you imported this generation mean data from here?
Sir, I have not imported. I just
Yeah. So
set the working
before
directory only.
before annotating, you need to import the data. Then you can annotate.
Okay, sir. Thank you, sir.
Yeah. The name. Alpha lattice. 1 minute. There is. Yeah. Exclude.
Restart the session and again run that code and it is done.
Done, na? Okay.
Yes, sir.
Okay. No problem. It's good
Earlier
then.
I was getting error.
No problem.
Thank
Okay. So half dialyl and QTL mapping we will complete tomorrow. Tomorrow is Wednesday. Okay. Tomorrow we will finish that. Then we will complete if any whatever is pending there. So one by one we'll complete that remaining things. Many are asking that TTC also. And if you want to study, as I already told you, that book name is there in chat box. So if you have, it's well and good. If you don't have, I will suggest that you should have. That book will give you a complete idea on the practical values of this biometrical genetics. That book is not that much big. It is very small book, but still on each and every aspect calculations are there. So easily you can refer that book. Okay. Okay. So we will
How
see.
can we get the book, sir?
It is available online.
Okay.
Yeah. Right from Amazon, like whatever the platforms are there, it is available.
Okay.
It is easily available. Okay, so tomorrow we will complete this part, and then tomorrow we will list out whatever is pending things. Then one by one we will complete it. Okay, okay. So tomorrow also 6:30. We will start at 6:30. Okay, thank you.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.